and you all can see me and hear me clearly, right? I'm audible.
Yes, sir.
Yeah. Okay. So if the network fluctuates in between, which it might be because I'm on a new network, on a new place, so do let me know. Okay. Very well. So how about you, Matthew? Let's start with recap, a quick summary.
Yes, sir. Sure.
Yeah. Where did we leave?
Yeah. So
Where did we start? Yeah.
we started with like the regulation of gene expression, and we were
Yes.
discussing prokaryotes the day before we were doing eukaryotes in the name of the
Okay. So let's
prokaryotes.
start from here, the one that you can see on the screen. Okay.
Yes. So in eukaryotes, there are mostly four kind of levels. So there is transcription level, then there is post-transcriptional processing, which happens after the transcription level. Then there is transport of mRNA from the nucleus to the cytoplasm, and then translation level. But in prokaryotes, it's a different case, because in the prokaryotes there is no need for the transport of mRNA from nucleus to cytoplasm.
Yes.
And
so there's only one way you can regulate, right?
yes, exactly.
Yeah.
So in prokaryotes, the control of the rate of transcriptional initiation is the major mode, as discussed. And then we discuss about accessory proteins. Yeah, and accessory proteins which involve positive regulators and negative regulators. And then we discussed about how they are involved in transcription. Yeah, and then we discussed that in a prokaryotic DNA, transcription is regulated by the regulatory proteins. And then we moved on to operons, which are operons, which are a special arrangement of a polycistronic structural gene in which multiple cistrons are regulated uh, by a common promoter and regulatory proteins.
Yes.
Yeah.
We discussed the example of lac operon, right?
Yeah, lac
so
operon.
Everyone is clear with the lac operon. Would you like to quickly tell about the lac operon as well?
Yeah, so basically there are two cases in which is one in the absence of inducer and in the presence of inducer. So in the absence of
First, before you begin, I think the way you should, if it comes in your exam, the way you should start writing the operon is always tell what kind of operon it is first. Lac operon is a negatively regulated operon because there is a repressor protein that always keeps the operon in a shut off state when the inducer, when the lactose is not present around. So basically you just have to write the logic first, then it makes easier to explain. And operon are for sure going to come in one of your exam, either the final boards or your school, but it will be there. It is a topic which cannot be left untested. So because it's also easy, but students find it difficult because they do not understand the logic. So the logic is all the genes that work in the same metabolic pathway or in the same pathway or for same process, let's say. Here the process is to metabolize lactose. So lactose is a sugar source, right? And it is not always present around in the surrounding of a bacteria, E. coli. So E. coli normally metabolizes glucose, get its energy. But if lactose is present around, then it has this set of genes Z, Y, and A. Z translates into beta galactosidase, Y into permease, and A into transacetylase. These three are enzymes. And all these three enzymes are required in the same pathway of lactose metabolism. So that's why they are under the same promoter and they form a operon. So this operon is negatively regulated, which means there is a repressor gene that codes for a repressor protein that always keeps the operon in a shut-off state because it's bound to the operator region. This is how you should start. Okay, Matthew? Yes,
okay,
continue.
sir.
Yeah.
Yeah. So the repressor mRNA creates a repressor, and then in the repressor, when there is an inducer, this creates Because there's inducer that creates two types of mRNA, which is the repressor mRNA and the lac mRNA, in which Z codes for beta galactosidase, Y
Yes, so
codes for
see,
permease.
anyways, yeah, that's a good point you raised. So the repressor mRNA will be produced all the time, okay, because
Yes,
it's a
sir.
negatively regulated one, and this repressor RNA is not regulated by any other thing. So it just has a promoter and this I gene. Can you see my cursor, everyone?
Yes, sir.
I hope. Yeah. So this promoter region, RNA pol can sit here every time. It just needs sigma to tell it where to sit, the sigma factor. So this mRNA, this blue one, will also be synthesized in absence of inducer and will also be synthesized in the presence of inducer. And it will also get translated because, you know, in prokaryotes, once it is transcribed, you cannot do anything. It will be translated because they are coupled. So the protein will also be formed in both the cases. Now the role here comes of the inducer, which is the lactose molecule in low concentration here. So when lactose is there, it binds to this repressor and inactivates the repressor. So in the presence of inducer, now the repressor cannot go and bind to operator region like it was in the absence of inducer. Yes, good. Yeah, finish it.
Yeah, and as I was saying, Z gene, the Z codes for beta galactosidase, Y codes for permease, and A codes for acetylase through translation. And then all three of them have their own uses in which beta galactosidase is involved in the hydrolysis of lactose. Permease allows the lactose to come, uh, as in the name permease, and transacetylates translates the acetyl group and
Transfers the acetyl group,
transfers the acetyl group
yeah, to the
and
lactose.
to lactose. Yeah.
Yeah, and then that acetyl group helps in its recognition through beta galactosidase, which comes and hydrolyzes the lactose and energy is produced and the motive is fulfilled. Okay. So you
Yes, sir.
understood they are actually working very close. So it's like if they are working very close, put them in the same operon so that whenever one gets transcribed, the other gets transcribed as well. Right?
Yes, sir.
Okay. It's like you need, stud shin guards both together if you want to play. So keep them in the
Yeah,
same bag.
exactly.
Yeah, exactly. Okay, so we have also, or Arpit has also joined us. Hello, Arpit. How are
Good
you?
evening,
Good
sir.
evening. Good evening. Good evening. Okay, so let's begin the
Sir?
new thing. Yes, Arpit, you have a doubt?
Sir, if like a question comes on lack of parent, sir, is it
Mhm.
necessary? Do we have to draw this diagram, sir?
I think there is no other way that to explain an operon if you do not draw this diagram. It's not at all a diagram. It's you made a rectangular box, make sections, write P I O Z Y A. It's not difficult.
Yes, sir.
You
My
just
teacher
have to.
directed marks for this. Sir, I had the test. Said yesterday, sir. And sir, like,
Okay, very very good.
sir, I wrote everything about this, sir, functions, proteins, everything. But sir, she only gave 1 marks out of 3.
Very good. She gave 1 mark.
Yes, sir. Only I've written like a whole
You
paragraph,
should
and it
so you
was not.
should thank her for giving that 1 mark. If you do the same thing in my test, or if I if I were your teacher, I would have not given you marks. See, the point is when some concepts can be explained verbally, right? But some concepts makes more sense. It's not like you cannot explain operons verbally. You can, but see, you are starting your science studies. You are starting your career in science. If you want to go further in biology, and this just makes things very clear. You know the sequence, right? Why I spend so much of time explaining processes? I can just give you notes that you can go and write in the exam, right? For Operon, for that matter, I did not write anything, but I made you write. I dictated you certain things, but I did not write anything on board, right? Because
Yes.
that's not the motive. Because you did the same in an exam and you got 1 out of 3. Now my motive is not to get my students 1 out of 3 marks. It is, if you make this and you mark, you understand now there is a promoter for a repressor gene which will always come first, then there will be a promoter, a operator, and 3 genes of the operon. Simple. What is the difficulty in this, right?
Yes.
But this tells that you know what we are talking about. If you can make a diagram, you know the answer, you know the system. So always use diagrams. Have you heard this quotation? a single picture speaks 1,000 words.
Yes. Sir.
It's said for a reason. I'll give you a 1-page summary. Suppose I'm giving you a 1-page summary of a beautiful scenery of Himalayan mountains. Versus I give you one picture of that beautiful Himalayan mountain. Which one will you prefer?
Sir, same.
Right? So your teacher didn't do anything wrong. Okay. Don't hold him or her responsible. Just learn that, okay, it's good that I did this mistake in a unit test so that again, when a big test come, I can score full, that like you did in the last one, right?
Yes.
So football and biology are same. If you know it, it does not give you the guarantee that you will perform every time. Don't make these small mistakes. I think Matthew will agree with me.
Yes, I do agree.
Yeah. So just keep your level up. Okay, Arpit?
Yes.
No need to worry. Don't worry much for the 2 marks, but I think you will do better next time because you know now. Okay, so today we'll start with human genome and DNA fingerprinting. We'll try to wrap this chapter and go to evolution because there are many interesting things waiting for us. Okay, before that, I just wanted to ask all 4 of you, Fatima, Matthew, Sumaira, and Arpit, that what is the next exam that you are having in your schools? Because see, when I teach, I plan classes accordingly, and I also keep in mind that if anyone, because sometimes schools do this weird thing, like I have seen in other batches, they will be conducting an exam where it will be chapter 2, 3, and 8. It happens. Some small chapter from some random unit they will just put in. So is everyone's syllabus on track? Anyone having an exam in September, October? Yes.
So we have completed biotechnology in school.
In school.
Yeah.
And so basically you will be quizzed. So when is your next exam?
Sir, it's in, I guess, late September. It's not sure.
Late September.
But
So
sir,
biotech.
the syllabus is too much. I know. Sir, this I know.
The syllabus for the school exam. So can you send me the syllabus?
Yes, sir, I will be sending.
Okay, so that and also the tentative date for the exam. Okay, Samaira and Fatima, what about you? Samaira, Fatima, are you there?
Yes.
Yes.
For us, they told in October, sir, midterm. But I don't know about this syllabus.
You don't know about the syllabus. So what's happening in your school right now?
So
that's a good way to know. Yeah. What are you studying in the school?
We didn't complete our genetics till now. Our vacation is going on.
What is going on?
Vacation. Is going on.
Okay.
To the
so anything that you have started or covered in school that we have not covered in my class? That is my question.
So we started evolution and we reached 7.8, I believe. Yeah, we reached 7.8.
So which is the next chapter? That's fine. Yeah. Evolution. And when is your exam, Matthew?
It'll come around October 1st or September last. We'll have a half yearly.
Very well. Yeah. So don't worry about me getting a little ahead or behind, but that's okay. But something which is entirely different, like biotechnology for Arpit. And what about you, Fatima?
Sir, it's easy.
Yeah, I know. It's easy. Operon, for that matter, was easy. You scored 1 out of 3. Okay. Sorry. Enough teasing for one day. Yes, Fatima, what about you?
So we also started evolution and we were going in order, but we didn't do the first and second chapter.
Okay, you didn't do first and second chapter in school. Okay, that's fine. But anything else that you have done in school and we have not started or we have not done yet?
No.
Okay, very well. Cool. Okay. So I'll plan. Arpit, then you please send me your syllabus and your tentative date. Okay.
Yes.
Okay, very good. Okay, so let's begin, people. So knowing everything till now, we have studied genetics both molecularly and also in chapter 5 the phenotypic and how the field started and where did it went. Now we are coming to two major applications of the knowledge of genetics that we have. So today I'm going to start with the topic Human Genome Project. Now what does the word tells you about Human Genome Project. So this project was a very, very ambitious mega project. It was the first project of its kind in biological sciences, which included a lot of other fields, in fact, opened a lot of new fields of research and study with such huge a budget. So Human Genome Project, the goal was.
I want hair like you. I want hair
Sorry?
like you.
Like me, Fatima.
Sorry.
Fatima, you're not. No, it's fine. It's fine. But who do you want a hair like? And now I'm curious. It either could be me or it could be someone else from.
It was my cousin's.
Ah, okay, cool, good. Okay, so Human Genome Project. Let's come to, if you want a hair like him, you get to know, you have to know that why you cannot get it now, because you're born and your genetics will not allow it. Okay, Fatima, so please focus here. So scientists have the similar kind of questions that Fatima had, that what decides our hair structures, eye color, everything like that. And how many genes do we have? No one knew how many genes humans have before this project finished. And there was an estimation, because humans were not the first. Of course, we cannot be the first organisms to do anything. We are the most evolved one. So everything has to start from basic organisms. So they started with yeast. They started with other simple systems, where they started sequencing their genome. Genome means the whole set from chromosome 1 to whatever number of chromosomes they have. They always sequenced only one copy, because the other copy is going to be a copy. It's a copy, right? It's the same thing coming from father. So in humans, if you have to do a Human Genome Project, tell me how many chromosomes will be required to be sequenced? How many number of chromosomes will be sequenced to sequence a complete human genome? Yes, Arpit. If you want to find the total number of genes humans can have, and if you have to sequence the whole genome of a human, how many chromosomes do you think you have to sequence? Am I audible, people? No?
Yes, sir, you're audible.
Oh, okay, thanks. So how many chromosomes we need to sequence to get the whole human genome? How many chromosomes do we have? Sorry, Arpit?
46.
46. Yes. How many do you think we need to sequence to get the whole human genome? To know all the genes, all the different genes, and the length of the whole genome, etc. Arpit, I think, gave an answer. You need to sequence 24 chromosomes. You understand how and why? 22 are autosomes, right? And it comes in pairs. So 44 chromosomes are just copy of each other. 22, chromosome 1 to 22 from mother and 1 to 22 from father. So any one side you sequence. And then X and Y are two different chromosomes. So we have to sequence both, right? Because they carry different genes. So 22 autosomes plus X and Y. That will be the whole human genome. Is it clear?
Yes, sir.
So it's not 23, it's not 46, it's 24, which if it comes in your entrance question, you will think that 24 is the least probable answer, but that is the correct answer. Okay. Okay, now write down that Human Genome Project. It's also known as HGP. Okay. So it was a mega project. When we call something a mega project, scientists do mean it. It was a project which was aimed, write down, it was aimed 233. Fatima wrote a number 233. What is 233, Fatima? Can you speak, Fatima?
So I'm typing.
Oh, okay. So write down in the meanwhile that Human Genome Project was a mega project which was aimed to sequence to know the sequence. Sorry, to know the DNA sequence. To know the DNA sequence of the whole human genome. To know the DNA sequence of the whole human genome, to find the total number of genes humans carry. To find the total number of genes human carry. I can understand you, Fatima. You know, work from home and study from home is difficult. It's okay. Everyone wrote?
Yes, sir.
Yeah. Now let's come to the logistics now. Why it was called a mega project. So how many base pairs does a human genome has? When I was when I was teaching you packaging of DNA, I told you about this. Do you remember? Humans, the human genome, haploid human genome, okay, and one copy has around 3.1 billion base pairs. 3.1 into 10 raised to power 9 base pairs. I think 9 is billion, right? Yes, 9 is billion. So 3.1, around 3, more than 3 billion base pairs were there to be sequenced. Now, can you imagine 3 billion base pairs? You have to know the sequence of each and every nitrogenous base, A, T, C, G, and so on, starting from 1 to 3 billion. And the cost which was associated was approximately $3 per base or per base pair. Now, can you imagine the amount which was required for this project, which was sanctioned for the project? 3 billion into 3. It was around more than $9 billion
Per
were
person.
invested for one human. Yes. And it was done once only. It was not like you are doing it for 10 persons. Because when you just need to know a human genome in its entirety, and you can do one thing. Instead of just taking it from one person, pool the sequence, right? Pool the DNA, pool the chromosomes. Do you understand? Because everyone is human, no? So it's better you take sample from multiple humans and then do it together. But it was coming out to be $9 billion. More than $9 billion were sanctioned. It took more than that. It is just on paper. Okay, this is just the cost of sequencing. You can understand when a project starts, there are a lot of other things, other costs associated. Yes. So of course it was going to be a mega project, but not only in terms of resources involved, also in terms of the output. The data that will be generated out of this project will be beyond human comprehension. Like for the first time, for example, when we will get to know all the 3 billion base pairs, you know how big is the data. So let's try to understand it like this. Let's say we want to store the data in form of books. Okay, and every book contains 1,000 pages. Okay, so if one page contains 1,000 letters, you understand, like ATGC, without
Yes.
any comma, without any punctuation, without any preface, summary, introduction, nothing. Just start from page number 1, put the first base there. and keep writing. No page number, nothing. It would be the most boring book ever published, but if that book is there with one page contains 1,000 letters and there are 1,000 pages per book, okay, then you will need somewhere around 3,300 such books to keep the human genome. Now, can you imagine it? It's a library in its own, right? And a very big one. 3,300 books. All the books are just sequence, nothing else.
It's
No
painfully
summary, no
boring.
preface. It's very painstaking a process. And now imagine after this data is generated, someone asks, can you tell me how many genes are there on chromosome number 1? Now you have to go through. How do we identify genes? Now you know that. What is the best way to identify a gene? Every gene has a starting point and an end point, open reading frame, right? Every gene codes for. So there should be a
Yeah, by
start
reading the
codon.
sequence.
Yes. So by reading the sequence, all you have to find in that sequence is a AUG, then billions of bases, and then in the end UAA or UGA or UAG. So you know that from here to here is one gene, right? Because this is start codon and this is end codon, right?
Yes.
Imagine you doing it in 3,300 books manually. Is it possible?
I don't think so, sir.
No.
Even if it's possible, no one will do this manually. So Human Genome Project, before it launched, the goals were big. In fact, Human Genome Project started rapidly, it developed. It was already started, like the field of bioinformatics was already existing, but it rapidly developed the field of bioinformatics. So one of the goals of Human Genome Project was also to improve the tools for data analysis, make computational tools. Now, doing something computational is not associated with biology normally in anyone's brain. So you understand $9 billion were just for sequencing. Then you have to develop new tools that can be used to handle this data. Then you will also develop a team, people, you will train people who can work with it. You can collaborate with industries and other areas which will help you with the technology and the working mechanism, right? It's not just biology happening. We are getting to know a lot. Yes. So first thing, you understood the project. What was the project and who funded it? Of course, the question, who was the mighty institutions who thought that, okay, let us do it? Any idea?
USA, the US government.
Yes. So it was a 13-year long project. Okay. It was a 13-year long project on paper, but it took more than this time. Whenever you make a project estimation, it sometimes goes beyond because you do not know what will happen when you are halfway into the project. So it was on paper 13-year long. It was started in 1990 and was supposed to end in 2003. Okay, so according to NCERT, this is the time frame, but you also must know that the last chromosome, which is the first chromosome number 1 in humans, is the biggest chromosome, largest chromosome, containing the most number of genes. Is it clear? So the last chromosome, which is chromosome number 1, was finished in May 2006. The sequencing finished in May 2006. So write down. It was a 13-year long project, which was completed on paper in 2003. So 1990 to 2003. And then some last bits of chromosome number 1, some last bits of chromosome number 1 were finished or were completed in 2006, May 2006. So if they ask you in entrance, which was the last chromosome to be sequenced among the 24 chromosomes, for Human Genome Project. It was chromosome number 1. Okay. You understand?
Yes.
Okay. So 22 autosomes plus X and Y. Now write down the goals of Human Genome Project. Oh, sorry. Again, I've not told you who funded it. So it was funded by first National Institute of Health. It's called NIH, USA. National Institute of Health. And second, Department of Energy. US Department of Energy. Okay, so these two bodies started it. Basically, both of them are under US government. But later on, when it was end, there was one more funding agency. It's known as the Wellcome Trust, UK. Now, the Wellcome here has double L, okay? Wellcome Trust is still a very big trust that funds research projects. And an interesting thing is, the lab which I was part of during my MSc was also funded by Wellcome Trust. Okay, so this is under UK, United Kingdom's Wellcome Trust. So these three started it, and then later on, I don't think it will be asked, but it was joined by countries like Japan, France, Germany, and China. So they are the, yes.
Sir, our country never funded any research. Like
we did Chandrayaan.
Oh, yes.
Mangalyaan. We have funded a lot of research. In fact, India is the fastest growing research industry. And talking about IT, every company is looking forward to now produce its technology in India due to two reasons. Indians are very hardworking and the labor is very cheap. We are a lot of people. We are 1.38 billion, right? Just like China. So the labor is very cheap here and things can get produced very, very cost effectively. And also, the IT technology of India, Bangalore, the Silicon Valley of India, Bangalore.
Yes, sir.
It's becoming very, very competitive to the actual Silicon Valley.
Damn.
Okay. Make in India, the Make in India initiative where Indians are starting entrepreneurship, making their own things. We are making brands now, for that matter. There are many Indian brands competing with established international brands from last 30 years, 40 years or so. You must be heard of a brand called Boat for
Yes, sir.
earphones, etc., speakers and all. It's an Indian brand. And they're doing quite well. UPI? Anyone knows UPI?
Yeah. The
Yes,
payment
sir.
service.
Who has found UPI? India. It's an Indian technology used all across the globe. UPI, an instant payment service, is an Indian product. Okay. And also many more things. And in space, we are doing quite good. Okay. Before, nowadays, everyone is very much awestruck or hyped by what Elon Musk is doing because he's making reusable rockets, right? It can just go, put a satellite in orbit, come back and launch. Stand again. And then again can be relaunched, put something in orbit, come back and stand again, right?
Yes.
Before that, and still now, it's not very cheap. It's going to be cheap when it will be used multiple times, because it's reusable. But do you know about PSLV? PSLV. It's a launch vehicle that is created by India. In a one go, it can launch as much, like the highest number is around 120 if I'm not wrong. We have managed to launch 118 satellites in one shot and put all in orbit successfully. So we made that launch vehicle, and then NASA and every other space agency, even private companies, telecom companies, other companies who need to put their satellite in orbit, they gave contract to ISRO to please launch our satellite, and we did. So 118 satellites in one go. I think the record is also
rocket.
In one rocket, in one launch vehicle. That's called one rocket. Is a launch vehicle. How many satellites you can launch through that launch vehicle in one go? That's the record. That's with India. So we're doing quite good in terms of research. Yes, we are still a developing nation, but we are developing at a good pace. I believe in terms of science, we need some support. Of course, support is always required, because if government of US of that time would not have funded this 9 billion project thinking, but they can do it because they are already developed. So some hindrance also comes from the society setup where we are. So if a country has major crisis in terms of poverty, you have to make food security first. Of course, food security has to be a priority. Even being a scientist, I will say that first you ensure for your people food, medicine, health, education. Then comes research and science. You understand? You cannot do research and science or space exploration at the cost of your own country getting, staying hungry or unhealthy. Yes or no?
Yes.
Yeah, but yes, we are doing quite good. Are you happy, Arpit, knowing that we are doing good?
Yes.
Very good. Thank you. Okay, so what was I saying? Yes, Japan, France, Germany, and China joined later onwards. So these are the major stakeholders. These two are the first, joined by Wellcome Trust, and then these countries also joined in some capacities. Now let's come to the goals of Human Genome Project. This will be a question in your exam. So they ask what were the goals of. Now the goals, there are two different things. One is goal, and the other is once you finish that goals, what we figured out. So they are called features, salient features or characteristics or outcomes of the project. So there are two things. Okay. So the goal is, before you start the project, you make goals. So write down. Goals of Human Genome Project. Okay. So first goal, of course, was to, okay, here I would like to point one thing out. In your NCERT, there are some very, very classical mistakes that they have made, which, they are not mistakes, but once you know science, you know that it does not make sense. So all of you have NCERT?
Yes.
Right now? Can you come to this topic? I don't know the page number, but can you please come to this topic, goals, Human Genome Project? It's towards the end of chapter 6.
So page number 118.
Thank you, Arpit. Page number 118, people. Can you see one of the goals? I don't know if it's the first one or which. They have written that one of the goal is to identify all the 25,000 genes.
Yes, sir.
Where is my phone? Just 1 second. Yeah, sorry. Yeah, can you see that they have written the goal was to identify all the 25,000 genes, something like that, a number?
Yes, sir.
But my question is, how did they know that there are 25,000 genes? If they have not done, like it's being done for the first time, right? Your estimate cannot be this precise. It can be, but in this case, it was not. So the goal was to know how many genes a human genome has. Okay. In fact, it was estimated that humans must be having somewhere around 1 lakh genes. 1 lakh to 1,40,000 genes.
Yeah,
must
but
be
then
there.
what they found out was lesser, right?
They found that there is approximately 28,000 genes, and it came as a disappointment. Because if you read more about Human Genome Project, there are many conspiracy
Yeah, because they
theories
thought that
around
humans
it.
are very developed and have more complex characters than other organisms, but turns out.
But that's true, right, Matthew?
Yeah, it is
Isn't
true.
it true?
Yeah, it
Yeah,
is true.
it is true.
But
So
they thought
what?
that there would be more genomes.
Yes, right. So earlier, scientists, geneticists always used to think that if an organism is simpler, it will have less genes, because it does everything is controlled by gene. So you don't need more gene if you're just a yeast. Then they found that butterfly, some species of butterfly can have around about 33 to 35,000 genes, butterflies, okay? And humans have 28,000 genes, less or more, give or take a little 1,000 up or down. So we have even less number of genes than a butterfly. What's happening? So clearly, but still we are more evolved than butterfly. That's true, right? Now we cannot say that it was a myth that humans are more evolved, we are less evolved. No, we are more evolved, but clearly it's not the number of genes then that decide complexity, right? You can be more complex with less number of genes. That is proven now. Once Human Genome Project was finished. You understand, everyone?
Yeah, sir.
Yeah. So it's not the, so write on the first goal. Let's first write the goals. To identify total number of genes, in human genome. Okay. Second was to also know the sequence or determine the sequence. Determine the sequence of all three billion base pairs. 3 billion base pairs that makes the human genome, one set of the human genome. The third goal was to store this information. Now, where can you store this information? I told you about books. That does not seem likely, right? It's not a good way. So if not books, yes. If not books, then computers. So even in computers, it was like you have to make a database. You understand? It cannot be just one computer. Yes or no?
Yes, sir.
Because now we have, you know, ROMs and hard drives till 5 GB, 10 GB. At that point of time, in 1990, how much do you think the capacity of computers? Do you know they used a computer to do moon landing? NASA used a computer to do moon landing. What was the RAM of that computer? What was the RAM in computers that did moon landing? Any
I'm
idea?
pretty sure it was like below 1 gigs.
One?
Below 1 gigabyte. I'm never for sure. It's like
Gigabyte?
Yeah,
Gigabyte?
yeah.
You are
Wasn't it?
gigabyte. Gigabyte was too big. Gigabyte
Yeah, I
just
said
came
it's
right
below
now.
1 gigabyte.
It's like you could have said it's below 10,000 gigabytes. It's like that only. 1 gigabyte was a big thing in 90. When did moon landing happen? Any idea?
1963.
69.
69.
1969. Yes. In 1916, the beginning, Kennedy said that by the end of this decade, we will go moon. And then there was a pressure because president said it. So they all just started doing this thing. Okay, we have to go
Then
to
he
the
got
moon.
shot.
We have to go to the moon. Yeah. Then he got shot. But they fulfilled. And without, before the decade end before 1970 started, they did it. And it was done by a computer whose RAM was in KB. Okay, GB was not thing there.
I expected at least megabytes, but
it was not even MB. Yes, it was not even MB, it was KB. So you can figure it out. I don't remember, but it was possible at that time. Now we think that, you know, even a mobile phone having 1 GB RAM is a headache. It does not work. It's not, because the point is everything is evolving. You know, even the technology is evolving. So one application that you run, it takes a lot of memory, random access memory that it can use to run. So applications are getting heavier, so you need more and more power to be generated through your motherboards and these chips. We are going towards making brains now, synthetic brains. So a computer is just the first step of a synthetic brain. Anyways, What did I say? Yes, store. So store the information, big information in database. So you have to make a database also. Then once the information is stored, what you would like to do? You have information, you have lots of information, but that's not the real science. The real science is what will happen now. It was just, you got the sequence, but now analysis part will be done, right? And that will be done by improving the tools. Write down, to improve or to develop. If the tools are available, you have to improve them. If they are not at all available, you have to develop them from the scratch. So to improve or develop tools for data analysis. And these tools are in silico tools, right? Softwares, basically I'm talking about hardwares and softwares. Tools for data analysis, because only a computer can help you analyze the data, which is so big, right? Today, this big a data is not considered a big data, you know, and it's not very, like it's just how many years? 1990, it's 2022. So just 32 years, right? In 32 years, now we are handling bigger data than this. But at that point of time, that's why it says whoever is the pioneer should get that respect, because starting and doing it is more important. Once it started, it will just keep evolving and developing. So at that point of time, it was a very big deal. 5. Yes, once you have developed the tools, now you also, the other goal was, so these are all the goals which were given by the authorities to people. So these goals were put out in open for citizen, and it's a very good thing everywhere, like even in India. All the science, all the research is done from whose money, who funds these research. So we say that NIH and Department of Energy funded it, but where did they get the money from? It's taxpayers' money, isn't it? Who generates money? Services and goods generate money in a country, right? It's people who are working who generate money, economy, right? Have you ever thought that a country cannot just keep printing notes? People, do you have ideas how does a country makes money?
Yes, sir, because if you keep printing notes, the value of the money will go down.
Yeah, it will become paper then.
Yeah, exactly.
Because that
It
money should
happened
have.
in Germany back in like 19, before the World War II.
Yeah, it happened in Spain. If you are right, if you're using the right thing, I think it was Spain you were talking about.
No, I was talking about Germany itself, sir, because they had a debt due to World War I. So what
and they just printed.
Yeah, they just printed notes.
Thought of getting rid of the debt, right?
Yeah, exactly.
But you know, if you study economics, so I was very keen in studying economics, and being a science student, you should also look at other areas, be more versatile. See, because I don't believe in, you know, becoming so monochromatic in your thought process that I can only do this and nothing else. That does not work. So in economics, there's this one line. When you start studying about money, the first line that I remember is money is what money does. That's the first line. So exactly, money is what money does. Okay. So in a country, it's all taxpayers' money that comes to the government back. Goods and services are generated. It goes back in the development of the nation. Then again, you avail those services, buy goods, pay tax. So it's like that. The more you will generate things, the more you will buy it. Money should keep rotating. Okay. If everyone just earns money and keep it in the bank accounts, the economy will not work. You have to be a consumer also. So this consumerism is actually important. Otherwise, what is the point of money? Okay. So you are always answerable. In a democracy, anyways, you are answerable to people for everything. But especially when you are investing as a country $9 billion into something, people will ask. If people will not, opposition will, and media will be there. So government has to justify. So these goals were there put to justify things. So they said that we will identify total number of genes, we will determine the sequence, we will store the information, improve tools, Fifth, we will also share the related technology with other sectors like industries where it can be put to good use for human benefit. So you have to show that, you know, at the end of the day, something is going to come out of it for you people, because it's being funded from your money. And so write down, fifth was transfer and share. Transfer, share technology, related technology with industries. With industries. Industries are very important sectors because they are the only ones whenever something comes. So every discovery, research is done in a lab, right? Yes or no?
Yes, sir.
Every medicine was found in a lab. Every drug was developed in a lab. But lab cannot synthesize it for the whole humanity, right? It has to be companies, industries, you know, like Ranbaxy, Cipla, these big pharmaceutical companies. So we know that what is paracetamol, but the lab who invented or discovered paracetamol is not the one making it right now. You know, it's industries. So everything that has to reach humans have to go through this large scale production that can be done by industries. And last point was to address issues. Of course, there will be questions. There were questions when they said that we want to go to moon. And if you see Kennedy's speech, if you hear Kennedy's speech, I think it's there. No, it's available, right?
Yeah, it is available, sir.
Yeah. So as a leader, he knew that it's very difficult, just because it was not just to, it was to an extent, let's admit that it was a race. It was a space race.
Yeah, it was
between
a space race between
superpowers.
Russia and US,
Yes,
and also
Russia
the
and
Cold War
US.
was happening at between that. So he wanted to prove that he's better.
Yes, so it was to fulfill, the testosterone power or the ego that we are the best. No, we are the best. So let's see who goes in the space first. And who won the space race? Like who won?
US did, right?
After the moon landing.
Yeah,
yes,
after
they did.
that.
Before that, who was ahead?
Russia,
It was Russia.
Russia, yeah.
The first satellite was Sputnik.
Yeah, the first person to go to space also as well
was also a Russian. Yes. So the next destination was, when you are losing in a race, then you say that, okay, we lost in a 100-meter race because we are a marathon runners. Are you up for marathon running? Challenge the opponent for a difficult task. Yeah. So they said, let's see who goes to the moon first. And Russia knew that, going to moon is not an easy task. So they didn't say anything. And America was like, okay, so they are getting on the back foot now. So let's just say that we will go to the moon by the end of the decade. And everyone was like, what? But then he very, very intelligently, was a very good orator. He joined it to the pride of Americans, every American everywhere in the world, whenever it will be known. So Americans were very happy. Yes, let's go to the moon. What is an ordinary American getting from going to moon? Nothing, right?
Exactly.
Yeah, it's just American pride, really.
Yeah, it's not like astronauts are going to bring one moon rock for every American. You will not even get a souvenir, right? It's just like they're telling you that let's do this in the name of nationalism and let's make America great again. The same thing, and it worked. Sentiments work in politics, and it should. That is that's drive sometimes science also and research also, and it did. And the moon landing happened. Have you ever thought that after 1969, why no human went to moon? Like they did. America, it was not just America went to moon once. America went to moon
they did multiple
6
times and they also failed. I think like 2 or 3 times.
They succeeded 6 times. They failed the first time. They failed the first time so miserably that the project was about to get closed because astronauts died in the cockpit.
Yeah. Basically, they got lost or something, right? Yeah.
The problem was earlier. Have you seen these space movies? Just a little detour, but I think it's very interesting that the doors always pop up towards the out.
Yes.
Yeah.
That was a later invention. Earlier, they have to be open towards inside. That was easy to make. But they didn't realize what in case of a fire. So in case of a fire, they try to make it as fireproof as it can. So there is this saying in NASA: every person who works in NASA is the ultimate nightmare is one thing: fire in space, colossal disaster. So you cannot do anything. So during the test runs, one of this cockpit, the first flight, the first project that was about to land on moon was not Apollo 13 actually. Neil Armstrong was not supposed to be the first person to set foot on moon. It was just his destiny that he became the first one. The first ones, the crew that was getting trained, there was a fire in that cockpit, and due to that, pressure built, and they could not open the gate now because you have to open it inside. You understand? So they could not save it, and NASA and the government was under so much of scrutiny and everything. They somehow still managed to do it again.
Yeah, sir, but Apollo 13 was after the moon landing, right?
Sorry?
The one you're talking about is Apollo 13, right?
Yes, Apollo 13.
Yeah, that was after the moon landing, because the moon landing was Apollo 11.
Sorry.
And that
Oh, first was Apollo 11.
Yes.
Okay, so that one, yeah, right. So Apollo 11 was not the first one to land according to the plan. So Neil Armstrong was not the first person planned by NASA who will set foot on moon. So the first 3 crew died during the test, because then they realized we cannot make the doors inside. It has to pop out in case of a fire or emergency, so that the astronauts can escape. So they paid a heavy price actually, to win this race. But after that, every country realized going to moon is like, it costs a fortune, but tells you nothing more than just we went to moon, right? Now is the age where we can explore space. We have technologies, we can get to know things, but about moon, after NASA's that venture, no country went to moon, like send manned missions to moon. They send machines, of course, but not human, right? And now again, the same kind of a mentality, the space race is beginning. You understand? Because a lot of private players are also in the competition now. It's not just
exactly,
NASA,
sir.
ISRO, and Russia. It's also Blue Origins and SpaceX and all these things.
Yeah, Virgin as well.
Yes, Virgin Galactic as well. Okay, so write down the last issue, which was the most important one and most difficult one for government, is to address the ethical, social, and legal issues related to the project. Okay, and it's mandatory for institutions and places like NASA, who are funded by the taxpayers' money, that whatever they figure out, whatever good or bad happens, they have to put it out in press within 24 hours. You know that? Otherwise, you can be held legally. There can be legal actions. So if something bad happens, you have to just tell it out to the world. But I think like there will be many things they don't tell us, but okay, cool. Anyways, so Human Genome Project the goals are clear to you?
Yes.
But we are not just studying it for its magnanimity. It's very big, it's a mega project, so let's study it. No, we are studying it for the science. So what happened? These goals were laid, many of these goals were fulfilled, and to do that, we are studying it for science, and science is in the methodology. So write down methodology. So what did they do? How was the human genome sequence? That is the part you should be more interested in right now as a science student. So there were 2 ways. Okay, in methodologies, write down. There were 2 major approaches. Because remember, there are 2 things they want to do, right? Find the sequence of all the bases, 3 billion base pairs. Okay. Second, find the total number of genes. Now, finding total number of genes means genes that transcribe into RNA. You understand or not? Only that region that transcribes into an RNA is called a gene. Yes or no? The region like a promoter region, which does not transcribes into a RNA, enhancer region, terminator regions, and other regions, they are not genes, right? But they are a part of DNA. So there were two major approaches. First was called EST. Write down. Expressed sequence tagging. Expressed sequence tagging. Okay, EST. Now, as the name suggests, expressed sequence, what does it mean? Only the sequence that is expressed. Expressed in the form of what? RNA, right? RNA,
Yeah,
then
sir,
protein. Yeah. So expressed sequence tag technique, or this approach, was to write down expressed sequence tags. This approach focused on identifying all the genes focused on identifying all the genes that can be expressed as mRNAs. All the genes that can be expressed as RNAs. Don't write mRNAs, just write RNA, because there are many types of RNA. You know that now, right? That
Yes,
can
sir.
be expressed as RNA, whether it is rRNA or any RNA. Yeah. So this is a directed technique, right? To find the number of genes. And in fact, finding the number of genes, just getting a number that it is around 28,000, give or take 200 up down, is easier. So EST founded the number of genes, approximate. The second approach was to find the sequence, the exact sequence of all 3 billion base pairs. Now that was a little convoluted. And the second approach was, write down, sequence annotation approach. Sequence annotation. This is also commonly in the field known as blind approach, blindfold approach. Write down. The blind approach is simply sequencing the whole set of genome. The blind approach is simply sequencing the whole set of genome. Containing both coding and non-coding region. Containing both coding and non-coding region. Okay, whether it codes for mRNA or a protein or does not code, and everything, promoter, terminator, enhancer, all regions, junk DNA, everything. Coding both, what did you write? Containing, yes, containing both coding and non-coding region, and then later finding the function of it. And then later finding the function of it. So blindfold approach says, do not segregate any sequence on the basis of whether it codes or does not code. Treat everything as same. Just get to know the sequence. Then after we know the whole sequence, we'll figure out which is coding part, which is non-coding part. You understand?
Yes.
Yeah. Now this is a little convoluted technique, and how it is done is, write down. Let's write down the points. So I'll just in sequence annotation. Okay. First, what do you need to do? If you have to sequence a genome is a very big thing, especially a human genome. It's very, very big. So you cannot sequence it in one go. You cannot hold it like a thread and keep reading the thread. You have to make fragments. workable fragments, lengths which can be worked biochemically. So write down for sequencing. First point, I'll just dictate, keep writing. First point, for sequencing, the total DNA from a cell was extracted. That's the first thing. Or isolated. Isolate the total DNA from the cell. First point. Second, convert it into random fragments. Just break it randomly. Convert it into random fragments. Now you must be asking, if we are breaking it randomly, how will we join it back, right?
Yes, sir.
That's called sequence annotation. That I'll tell you. So first take the DNA, make random fragments of smaller sizes. Okay, smaller sizes, because it's a technical limitation. We cannot read a DNA, we cannot sequence a DNA beyond a particular size. So make fragments of smaller sizes. Then third step, clone the fragments. Clone the fragments in a suitable host. Clone the fragments in a suitable host using suitable vectors. I'll explain this point, just write it. Using suitable vectors. What is cloning?
Making
A
a copy.
replication of something.
Yes, Fatima, what were you saying?
Making a copy.
Okay, same thing. Both of you are correct. So cloning is just making multiple copies of that one fragment that we have created. Now, you could have asked, take multiple samples and break randomly. But see, we are breaking randomly. So when it's randomly broken, and when you clone, we want that random fragment to be cloned multiple times, but it should be the same fragment. So for that, you need to clone it under inside different hosts. Now, these hosts, there can be two kinds of hosts, because you have to clone a DNA fragment, a small fragment. So you can do it in two things. One is bacteria. Okay. One host can be bacteria, and the other is yeast. What is the similarity between bacteria and yeast? Chapter class 11th. What is the similarity between a bacteria and a yeast? That both are unicellular, right? Remember, yeast is a unicellular eukaryotic. Yes, the only unicellular eukaryote. Bacteria is prokaryotic, so of course it will be unicellular. But yeast is eukaryotic, bacteria is prokaryotic. Okay, there's a difference. So if you're cloning something, some sequence in bacteria, it's done by, the vector is BAC. This is the vector. The full form is bacterial artificial chromosome, because it's human made, something that can express itself in bacteria and get replicated. BAC is bacterial artificial chromosome. This is the vector. Vector is just a, you know what is vector from physics? Vector. Yeah. Mosquitoes are vector for malaria, malarial plasmodium, which carriers basically, right? So this is the vector which we can use in bacteria. If it is yeast, we use YAC, same, yeast artificial chromosome. Okay, is it clear, everyone? What are these two things? They are the cloning hosts. So what step was it? I forgot which step we are on. Cloning is what step? Third, yes.
Yeah,
so you have to do cloning in suitable host using specialized or suitable vectors. Then what will cloning do? Continue. Cloning results in amplification of the sequence. Sorry, amplification of each fragment. Okay. Then next point, next fourth step, sequence that fragment using Sanger sequencing technique. Sanger sequencing. Now it is not in your syllabus, but if I find time, when I will start biotechnology, I'll talk about this. Okay, Arpit, so it's your job. You have to make me remember that, sir. Let's also discuss a little bit about Sanger sequencing. Okay, during biotechnology. So there was a scientist called Frederick Sanger. He discovered this technique through which you can actually read each base. How will you know that which is the base in this nucleotide? That's called Sanger sequencing. So then fourth step is sequence that fragment using Sanger sequencing. Okay, now till here it was easier. If you're thinking that this was difficult part, no, this was the easy part. Difficult part is now, because remember we fragmented it randomly, and to get the sequence back you have to join it reliably in the right orientation. And how will you do that? Any idea? The sequencing is done automatically. It's an automated machine that works on the principle of Sanger sequences. So we have sequenced till now. Now the point is to put the sequence in right orientation. Any idea how can we do that? Because they are randomly fragmented, there is one logic that we can use. If you fragment something randomly, there will, and if you are fragmenting more than one sample, then of course when you will get all the results back, you will see some overlapping sequences in different fragments. Yes or no?
Yes, sir.
Let me explain you how. Let's say this is one sample and this is another sample. This is the basis of annotation, DNA, this whole process, sequence annotation. So let's say here it had sequences A, B, C, D, E, F, G, H, I, J, K, L, M. Okay. Same thing is here. A, B, C, D, F, G, H, K, L, M. Okay. Now you are randomly fragmenting. Okay. Is it clear? Let's say we are fragmented. So this is one fragment, right? This is the other fragment, a big one. You understand? Randomly it can be, all will be not be of the exact same length, no? You understand? Are you understanding? Here A, we know that A and B are together because this fragment has A and B together. Everyone on the same page, say yes or no? We are doing research here. Okay. Yes, then we know that C, D, E and F are together because of this fragment. Okay. But what we do not know is that this C comes after B or before B. This fragment should be second and this should be first, or it should be the other way around. It could be anything, right? But here the random fragmenting helps. Look here. Through this fragment, we will come to know that A, B and C are together, right?
Yes, sir.
So we know that C has to come after B, and everything after C, D, E, F will of course come after that. You understand? So this is the logic. This is called annotation of sequences on the basis of some similarities in the fragments. And because we are making thousands and lakhs of fragments, it's not that difficult to do, but you just need a stronger computational powers, and their computers come into the picture. Do you understand, everyone? Suppose you are confused.
How do you make sure that it belongs to the same sequence?
Because we are just taking one sample.
Oh,
we just
okay.
take one sample. It's human sample, so we know that it belongs to a human sample. That's all. There are 24 chromosomes. So first you will start with, let's say, chromosome 1. You will make random fragments, do the sequencing, annotate it, and make a full. It's like solving puzzle. You can understand, no?
Yes,
from
sir.
This fragment, you know that A and B are together, but from this fragment, you know that C comes after B. And from this fragment, you know that after C, there is D, E, and F. So, and if C comes after B, then this should be the sequence, A, B, C, D, E, F. We got to know this, right? Everyone easy peasy, or are you confused?
Yes, sir, understood.
Fatima, Samaira, Arpit?
Yes, sir.
Very good. So that's what was done. So write down. Next point, the sequences were then arranged. These sequences were then arranged on the basis of some overlapping regions, like this CD region. This is overlapping region. Here also they see F here, this is overlapping region. So based on overlapping regions present in the sequence. Okay, so write down in the end, just to justify, write down one last point. This alignment was not possible manually. This alignment was not possible manually. So specialized computer programs were developed for this purpose. Specialized computer programs were developed for this purpose. And now write a star point. So Human Genome Project, this is the last point. Human Genome Project also led to the rapid development of a new field in biology, or a new area in biological research called as bioinformatics. These are called computational biologists. They do biology but in computers. Okay, so someone from a computer background, a computer scientist or computer engineer, if he wants to switch to biology, we are very accommodating people. Biologists are very welcoming people. Come, you are physicist. We made Mendel come in, become the father of genetics. Seymour Benzer come in. We made mathematicians come in, because every expertise can be used to understand life and to study biology, right?
Yes, sir.
Yeah. So if you are studying biology, just don't like the subject, be proud that you are reading this subject. Even if you do not, whether you be a biologist in life or not, whether you become a doctor or not, whether you pursue biology later or not, but you knew this subject, you read this subject, you discussed it with a teacher, that's a good thing in itself, right? So everything is clear about Human Genome Project?
So can you repeat the fourth point?
Yes, this is as difficult as sequence annotation for me. What was the Fatima, can you do this for me, please? Because I don't remember what was the fourth. What is the fourth statement, Arpit? Where does it start? Which line?
Sir, I didn't write, sir.
Okay, what was the last sentence you wrote?
Sir, clone fragment in a suitable host using suitable.
Okay, so after cloning, anyone can repeat it, please? After cloning, the fourth point.
Cloning hosts, we wrote bacteria and yeast, and then we wrote the point cloning results in amplification of each fragment.
Cloning results in amplification of each fragment. Okay, Arpit?
Okay, sir.
And then after that, Fatima?
Sequence that fragment using, we wrote about the Sanger sequencing.
Yes. Did you write that, Arpit? Sanger sequencing thing?
Sir, wait, I'm writing the first book.
Okay, okay. I think Matthew's voice is very clear. So Matthew, can you repeat the fourth point?
From
It was clear, but like she went too fast.
Yeah, okay. Arpit, Matthew, can you just repeat?
Yeah, so we wrote cloning results in the amplification of each fragment.
Cloning results in amplification of each fragment.
Yes, sir, written.
Done?
Yes, sir.
Okay, very well. Now, come to NCERT. The last thing I'm giving you, open your NCERT. before DNA fingerprinting starts, there are features or characteristics. So it might be a handing. Either it is feature or characteristics of human genome. Let me also pull it out for you if I can.
It is salient features.
Yes, that one, same. Which page it is? Can you tell me, please,
120.
Fatima? 120.
100 and yeah.
Okay, just let me go to online book. 120. Yes, I got it. So thank you, Fatima. Yes. Yes. Can you all see this? Let me make
Yes.
it a little big. Is it visible to all of you now? Salient features of human genome. So there are 9, but basically this 2 are actually the same things. But I just want someone to just read this feature. This is the last, and we'll finish human genome today, human genome project. Yes. Can you read it for me, Matthew? Because your voice is very clear.
Yes, sir.
So this is after it was done. What we found. What was
Yeah. Some of the salient observations drawn from Human Genome Project are as follows. Number 1, the human genome consists of 3164.7 million base pairs.
Yeah, this is an easy number. Just remember this: 3.1 billion base pairs.
Yeah. The average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases.
Can you imagine? The average is like total gene, total number of sequences divided by total number of genes, right? So the average gene sequence came out to be 3000 bases. But the biggest gene, which is dystrophin, human dystrophin, is 2.4 million bases itself. So one gene is 2.4 million bases long. Okay, so it varies greatly. Okay, continue.
Yes, sir. The total number of genes estimated at 30,000, much lower than the previous estimates of 80,000 to 1 million, 1,40,000 genes. Almost all 99.9% nucleotide bases are exactly same in all people.
Yes, right. We will discuss more about it in the next class, where we will finish DNA fingerprinting and start evolution. Next.
The functions are unknown for over 50% of the discovered genes.
Because we just discovered, we don't know what they do. Still, we do not know what every gene does. Okay, it's just 32 years.
Still, there has not been any research on that, or has research not found any?
Lots of research has been done, lots and lots, but still we do not know what every gene in our body does.
Oh, okay, makes sense. Less than 2% of the genome codes for proteins.
And they are called?
Cistrons.
Now you know it, right?
Yes.
Protein coding genes are cistronic regions, yes.
Repeated sequences make up a large portion of the human genome.
Yes, these 2 points are very important for tomorrow's class as well, because I will use this. So please go and also read about VNTRs today. I'm giving you this homework. What are VNTRs for tomorrow's discussion? Yes.
Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution.
Yes, they have a lot of function. Now we are finding their functions, about some of which I will discuss with you tomorrow. Okay. Next feature.
Chromosome 1 has the most genes, 2968, and Y has the fewest, 231.
Yes, and chromosome 1 has the most genes and was last to be completed. May 2006.
6.
Yes. NASA.
Scientists have identified about 1.4 million locations where single-base DNA differences, SNPs, single nucleotide polymorphism, pronounced as SNPs, occur in humans.
Along with VNTRs, also please read a little bit about SNPs. If you can understand something, well and good. If you're not, then don't worry. We'll discuss it in the next class. These two are important things for the next class. Yes.
This information promises to revolutionize the processes of finding chromosomal locations for disease-associated sequences and tracing human history.
This is already being done. So even labs are actually using these SNPs to figure out cause of many genetic disorders. Okay.